Alright everybody, it's time to talk about meiosis. On our last video we talked about mitosis, which is the division of body cells that produces cells that are exactly the same as what you start out with. Meiosis is the production of sex cells, which produces cells that are genetically different and have half as many chromosomes as what you start out with. So these two types of cell divisions are different, but they share a lot of similarities. And the first similarity comes in the beginning. When we look at an organism that is diploid, it has two copies of every chromosome. So we start out with a cell that's very similar to what we started out with in mitosis. We have two copies of every chromosome, one from each parent. And just like in mitosis, this cell is going to make copies of every one of its chromosomes during interphase to prepare for cell division. So far things are looking just like mitosis. The difference is that as this genetic material starts to supercoil and become visible as chromosomes, the chromosomes are going to pair up in what's called a tetrad. So these things called homologous chromosomes, which are chromosomes that have the same genes on them, but not necessarily the same version of those genes. Remember last time we talked about the genes that you inherited from your mom, or the chromosome that you inherited from your mom, and the chromosome that you inherited from your dad, with all of the genes that you inherited from your dad on it. Those are called homologous chromosomes, and they will pair up into what we call a tetrad. So by the time we get to prophase 1 of meiosis 1, we have homologous chromosomes paired up as a tetrad, nuclear envelope dissolves, and this very, very important thing called crossing over occurs, where pieces of one homologous chromosome are going to exchange with the same piece of its homologous chromosome. So while this is going on, you can imagine these chromosomes changing from being all red and all blue into a chromosome that's red and blue striped. And that process is called crossing over, and it allows your chromosomes to shuffle up the genes so that you're not passing on all of the genes from one parent or all the genes from another parent. You're passing on a mixed up or a shuffled up version of the chromosomes that you inherited from your parents. So that's what happens during prophase 1. Metaphase 1, the homologous chromosomes line up along the midline of the cell, all right, which is different than metaphase of mitosis. If you take a look at the mitosis video, you'll see the big difference there. We have homologous chromosomes lining up along the midline of the chromosome during metaphase 1 of meiosis. Anaphase 1, the tetrads separate. The chromatids are not separating. The tetrads are separating. So these chromosomes are going to opposite poles of the cell. So if you do the numbers on this, you still have 6 pieces of DNA, just like in mitosis, but the state of that DNA is very different during anaphase 1 of meiosis compared to anaphase of mitosis. So anaphase continues as long as these guys are going to opposite poles of the cell. Telophase 1, this cell is going to divide into 2 separate cells. New nuclear envelopes are going to form around these chromosomes. They're going to unravel. No replication of DNA occurs again. I repeat, no replication of DNA occurs again. So we basically go right into supercoiling, dissolving the nuclear membrane, and we're back in prophase 2 of meiosis 2. All right, so we've got 2 cells here that have not made another copy of their DNA, but they're going to go through another set of divisions. And that's one of the big hallmarks of meiosis, is we have 1 replication of DNA, but 2 cell divisions. So this would be prophase 2. Metaphase 2, they line up along the midline of the cell. All right, and keep in mind we're looking at 2 separate cells here. Anaphase 2, these chromatids are going to separate to opposite poles of the cell. All right, these chromatids are going to separate to opposite poles of the cell. Telophase 2, this cell divides into 2 daughter cells. This cell divides into 2 daughter cells. So we end up with 4 gametes that each have half as many chromosomes, n number of chromosomes. In this case, it's 1, 2, 3. One of the big, one of the medium size, and one of the small. Different combinations though. And in males, this would result in the formation of 4 sperm cells. In females, it would be 1 egg cell or ovum and 3 polar bodies. So that is meiosis, which differs significantly from mitosis. Thank you very much for listening.